Welcome to today's USP and Pharmatech webinar, specific aspects of the EU GMP Annex 1 revisions. Thank you for taking the time out of your busy schedule to join us. Next. Presenting today is Mr. Mark Mitchell, Principal Consultant at Pharmatech Associates. Welcome and thank you. I will now hand the presentation over to Mr. Mitchell.
Welcome, everyone. Today we're going to be talking about certain specific aspects of the EU Annex 1 revision, which was just published last September. This is a rather significant revision. The previous revision, published in 2007 and actually took effect in 2008, was only 16 pages long. The new revision, published last August, is 52 pages long and also includes a glossary. Some of the major highlights, some of which we'll be talking about today, it now includes what's called a comprehensive contamination control strategy requirement through a appropriate reorganization of the information. Now makes it very clear what is meant by classification, qualification, and monitoring of clean rooms and clean air systems. There have been some revisions to the particle and microbial limits table, which we'll speak about. There now is a requirement for what is called PUPSIT, which is the pre-use post-sterilization integrity test. There also is a new requirements around lyophilizer loading and unloading. This is the one aspect of the change to the Annex 1, which has a longer implementation time. It actually has a 2-year implementation time. And also there's some revisions to the requirements for container closure integrity. As mentioned before, the implementation time was 1 year from published for most aspects. That timeline is coming up to August of this year, which is coming up fairly rapidly. And the only aspect which has got a 2-year implementation, which is August of next year, is the lyophilizer loading and unloading. These are the particular subjects that we'll be talking about in this presentation. Unfortunately, with such a large revision over encompassing a rather large, significant number of topics within Annex 1, we can't cover everything, but these are some of the highlighted areas which we'll be talking about within this hour. First, we'll be talking about the contamination control strategy, what we mean by classification, qualification, and monitoring. We're going to talk about PUPSIT. New sections have been revised and added based on barriers and other advanced technologies such as single-use systems and closed systems. And finally, we'll have questions and answers. So what do we mean by this new terminology, contamination control strategy, or a CCS? What the Annex 1 is trying to do is indicate there should be a holistic approach to define all the critical control points to minimize the risk of not just microbial contamination and particles, but also pyrogens and endotoxins. So they want to talk about what is being done across the organization about all these particular potential contaminations and what controls are being put in place. So this is something which is implemented across the entire facility to assess effectiveness of all just various different aspects of control, whether it's the design of the facility itself, what procedures are in place, what technical aspects are being done in terms of equipment and systems, and also organizational. So we've seen this as part of Pharmatech when we perform audits in aseptic facilities. It isn't always clear where the responsibilities lie for control of contamination, such as microbials and particles. it may not be a single person, it may be multiple people in the organization, but frequently it is not clearly defined in any document as to who is responsible for what aspects of this control strategy. It's made quite clear in Annex 1 that there needs to be a periodic management review for effectiveness. So there would be an expectation from any auditor to be able to say, we want to look at your periodic management review, whether it be monthly or quarterly or whatever, or even annually. And we want to see, are you looking at your contamination control strategy? What events did you have over the last period? What CAPAs, what investigations, what breakdowns of control did you have? And have you updated your contamination control strategy in order to deal with these events? Whether it actually exists as a single document is still up for debate, but certainly there should be a document that you can pull, which is some sort of summary or reference or something that basically makes it very clear as to where all this information lies if it doesn't lie in just the one document. Again, we have seen in Pharmatech in audits where we go into an organization and we ask for a very simple litany of different controls, such as your cleaning strategy or your personnel gowning. And it takes some time to pull all those documents together because it seems that they're all spread out in different areas of responsibility and no one has an overall view of it. That I think is what the intent of the contamination control strategy is to provide a document where basically you can pull out and say, yes, this is all the different aspects of contamination control that we have. And this is all the different ways we do it procedurally and justification documents and whatever. And not to overlook, but also, how did we come to these conclusions of simple things like, why do we clean as frequently as we do? Why do we do particular monitoring points in our clean rooms and such? How was that determination made? What was the assessment? Was there a risk assessment? Is there data to support that? Was there a justification document? Frequently, these are scattered throughout the organizations and different responsibilities. And I think the intent of the contamination control strategy is to kind of put all those pieces into one place so they can be easily retrieved. Within the Annex 1, this is kind of the list of things which is to be considered to be included in the contamination control strategy. So design of plant and processes, process flows, layouts, particular elements of the design of the plant in terms of flooring and ceiling and such to provide for better contamination control. Premises and equipment. Is your equipment designed for control of contamination? Personnel, both their hygiene and gowning and such. Utilities that support your contamination controls, such as your HVAC systems. Raw material controls, a lot of particulate matter that ends up in our drugs. Is basically coming through some of our raw materials, both microbial and particulate. Containers and closures. Key suppliers and vendors approvals is very important. Do you have quality agreements in place with these particular suppliers? Outsource activities such as sterilization. How do you ensure when materials go out for sterilization and they're returned that they're maintained in a sterile state? I can give you one example that we found that somebody was basically using Ziploc bags to store their materials that are sterilized, so that when they were returned, basically they could be easily opened, but you're not providing great assurances of that bag has not been opened during either during or after the sterilization process. Your risk management, your process validation, all your sterilization processes, their validation, the preventative maintenance for the equipment that provides contamination control, HVACs, autoclaves, etc. cleaning and disinfection programs, monitoring systems, whether they be manually done or an automated total particulate monitoring system. How are your trends and investigations and CAPAs integrated into your contamination control? Are they flagged in a particular way so that in your CAPA or your investigation systems, they can be pulled? Trending is a very important aspect that's called out for in the new Annex 1 revision. There is an expectation of maintaining trends on the various data that you collect for both particulate and microbial contamination. And of course, continuous improvement strategies. Are there periodic reviews and potential improvements of the contamination control strategy? Now, there have been, I know that there's been a recent publication from the PDA for contamination control strategy. I haven't reviewed that as of yet. Haven't been able to get a copy of it yet, but so there is obviously some industry assistance, which is going to help in helping you put together a contamination control strategy for your organization. Because of the way Annex 1 has now been reorganized, I feel it's actually much clearer that these particular aspects of clean room and clean air equipment, in particular classification, qualification, and monitoring, they've been defined very clearly now so that we understand what the differences between these three different aspects are. The first aspect, classification, what we're talking about is really the application of the ISO 14644 series, essentially the clean room standards. So what we're talking about is this is applying the standards to measure the total particulates in a particular clean room. That starts by applying the formula for determining the number of sampling sites based on the square footage of the space per the ISO. So you grid out the room. You actually, within each grid, you put a portable particle counter. That portable particle counter is designed to measure 1 cubic meter of the air over a fixed time that's defined by the ISO to make a measurement of the particles in that particular space of the grid. You have to measure under this Annex 1, as we understand, we have to measure both at rest, which means all equipment in place but not operating and personnel not in the clean room, and in operation for both 0.5 micron and 5 micron. That's the requirement under Annex 1, although, ISO 14644, of course, is designed, for all sorts of kinds of clean rooms. It is not specific for pharmaceuticals, so it, of course, includes all sorts of different particle limits. Those particle limits are not of interest in our pharmaceutical industry. What we apply is either the FDA standard, or in this case, the Annex 1 standard from the EU, which now requires 0.5 and 5 micron. The criteria for this is in the Table 1 of Annex 1. And they indicate that, of course, if you're doing an initial qualification, you obviously would not be doing product because you'd want to classify that and qualify that facility prior to actually putting product in. So you would do it during a simulation, a water run or something like that. It also indicates that if you're doing a reclassification, you may be doing that reclassification during an aseptic process simulation or a media film. Now, when we talk about qualification of a clean room, that initial qualification of the room of a clean room, and of course, within the qualification of a clean room, you have to classify it. So yes, classification is now part of qualification. Qualification also includes the filter integrity tests for the HEPAs, air velocity and volume tests, pressure differential between the different grades, airflow direction and visualization. These are our what we used to call smoke studies, but they're basically some method of visualizing that the airflow is appropriate between the different grades. The microbial tests, which are listed in the Table 2 of Annex 1. Also, within that qualification, it's clearly stated now, temperature and humidity. Now, of course, the Annex 1 does not indicate what is expected for temperature and humidity limits. That's determined by your own product and personnel requirements. Recovery testing, and where appropriate, contamination leak tests. Now, this is an important aspect that's been made clear within the Annex 1, is now the requirement for a periodic requalification, which is every 6 months for grade A and B, and every 12 months for grades C and D. The Annex 1 indicates that those aspects that are required for requalification are classification, the HEPA integrity test, the air velocity and volume test or recovery test, depending on the grade, and the pressure differentials. Now, many of these are pretty common to us for who do and maintain clean rooms, such as the HEPA integrity and air velocity and the pressure differentials, for example. However, it is not necessarily always been done that you perform the classification per the ISO 14644 as part of a requalification that's done every 6 months or annually, depending on the grade. So that's a little bit more open to interpretation. I think industry has to deal with that a bit, because it is not a simple task to go in and bring all these portable particle counters in and perform this classification with that frequency. And this is what the Annex 1 states. So we have to decide that is this going to be a full classification per the ISO 14644? Is it going to be some sort of subset? That is still open for debate. So we will have to see how that is particularly interpreted. Finally, I want to talk about monitoring. Monitoring is what we do essentially every day when we do production. We need to do certain monitoring for our contamination control. And the main aspects of that are the environmental and process monitoring, which include, of course, total particles, our viable monitoring, both for the environment and the personnel. We're going to maintain and monitor temperature and humidity, and potentially other aspects of our clean room. We may monitor the temperature differentials between grades, between rooms, monitor the pressure, for example. And then they include within the monitoring section the aseptic process simulation. They consider this aspect of monitoring, but for those who do APSs, that's a fairly routine aspect. These aspects are to be used as part of routine batch release. So when we release batches, there's now a clear expectation that we will review the environmental and process monitoring results for those particular batches before we release them. So if we have events during our batches, if we exceed certain limits, we have to have investigations appropriate to what those excursions are. We need to use risk assessment methodologies to determine location, frequencies, and methods. So if we are going to say we're going to have certain locations for these monitoring probes, whether they be a automated system or whether they're added manually or whatever, we need to define those locations, frequency, using a risk assessment. Now, of course, there's more restrictions in terms of the critical areas, the grade A area of what's expected in terms of locations and frequencies, and we'll mention that in a minute. Now, to be clear, the monitoring limits for total particles and viable particles are different tables than the classification limits, and we'll go into that in a moment. There's also an expectation, particularly for the lower grades, to establish, based on either risk assessment and historical data, to establish your own alert and action levels, and to have appropriate alarms for those levels. So it is not going to be appropriate for you just to simply use the max tables that you see listed in Annex 1 as your action limit. Because as far as the EU is concerned, if you're already beyond the max of the table, you're already non-compliant. You want to have an action prior to that non-compliance. You have to determine yourself how you set those particular levels, and how you alarm to those levels and respond to those alarms. There's also a strong expectation of trending analysis, right? So you can't just have the data collected. You can't just simply say, well, we release it batch by batch. You need to actually have some sort of trending program. Trending programs for microbial are pretty common. However, it is not as common to see a trending program for total particles. There is an expectation for both. The revisions to the Annex 1 table are shown here. I've indicated with the circles particular areas that have been changed. The big change has been the 5-micron limits for grade A and grade B have now been changed to not specified. That means that you don't necessarily have to have limits for those in classification unless your contamination control strategy has set those limits. We'll go into a minute in the next slide when we talk about monitoring. What the challenge has always been with these limits that are very low numbers relative to a cubic meter of air. There's also been a slight adjustment in these other levels. The 2930 that we're seeing used to be 2900. This is just simply a minor adjustment because now it aligns more appropriate to the limits that are set in the current ISO standard. Now note that this table looks similar, but is actually different. This is table 5. This is the monitoring limits table. And so, whereas before we didn't have limits in the 5-micron and the grade A and the grade B at rest, now suddenly we have limits again. So, oddly enough, we don't necessarily classify to a limit for 5 microns, but we have limits for monitoring. The reason is the intent is that over time, monitoring the 5 microns is a good indicator of a deterioration of a process or a clean room for particulates. So over time, as we look, the 5 micron has actually been a good indicator. The 0.5 micron may go up and down, and it may trend up, but the 5 micron, if you're suddenly seeing a lot of 5 micron particles, that's an indication of a problem. They did add a section, which is very important, which says that 5 micron particle counts in grade A may be considered false counts due to electronic noise, stray light, whatever, if not regular or consecutive. The reason this is in there is that there's a certain practical aspect of how we measure and the frequency we measure air when we do monitoring. The most particle counters are designed to collect air at not a cubic meter per minute, but as a cubic foot per minute. And the reason this is because a cubic meter is 35 times faster. And that's actually a rather disruptive velocity for a particle monitor. So it has been determined that a cubic foot is the more appropriate cubic foot per minute is the more appropriate velocity in which you do particle monitoring. The problem is if now you try to measure and convert from a cubic meter to a cubic foot. So within a minute, in a cubic foot, you're essentially you're at a fractional particle, right? You're dividing 29 by 35 to get to a cubic foot. So okay, there's a practical thing. You can't measure a partial particle. So does that mean when I see one particle in 1 minute, do I fail? Well, no, it's more complicated. You have to determine methodologies, whether you're seeing it over multiple minutes. And yeah, so just remember, this table is not based on minutes. It's based on volume, and how much time it takes to collect that volume over time. So there's also needs to be read more carefully, but there's also interpretations of people that use what's called a rolling count, where they try to take a count over 35 minutes to determine these limits for 5 micron, for example. And there is a very explicit description of how to interpret that within the new Annex 1. Note here that there are limits both for in operation and at rest. So there's an expectation that after your operations have completed and your personnel have left, that if your room now goes into a rest state, that within a certain amount of time, called the cleanup period, that you will now get to that at rest state. But no, you have limits both in operation and at rest. You can't ignore during monitoring your at-rest limits. So you have to figure out how to do that with alarms and appropriate responses and things like that. Again, I'll mention that there's an expectation, particularly for like 0.5 micron and higher or lower grades like C and D, to not use this table as your action limit. They're really, for like C and D, these limits are way too high for most typical clean rooms. Your historical data is much tighter than this, and that should be used for your trending and your action limits, really to try to show that you have a certain level of control of your clean room, and does it deteriorate over time. I'll briefly mention the microbial tables here. These are set different tables. These are for microbial limits. Table 2 is the qualification table. Table 6 is the what I would call the monitoring table. Pretty much the data is the same. It's just that the monitoring table includes the personnel monitoring for grade B and grade A. The main change between the previous version was that it used to say less than 1 CFU, and now it says no growth. It's trying to make it very clear that there's an expectation that any growth within grade A requires an investigation. There should be no microbial growth detected in any of these methods in grade A. And again, obviously you can't be more stringent for grade A because it's already no growth. But for grade B or C, particularly more C and D, more stringent alert and action limits really should be established. Generally in history, you're going to see much better performance in a clean room, even a C and D clean room, than what you're seeing here in these particular limits. So these are some particular aspects also of importance that have been mentioned in the revision. Some of these are new, some are not. For grade A, total particles and viable monitoring is for the full duration of critical processing, which was correct from before, but also includes equipment assembly. We've seen this frequently in audits, that a lot of times that the data for monitoring for total particles and even viable monitoring is not done until after the equipment is fully assembled. Well, as we understand, this is an aseptic assembly. The interventions of putting things together can contaminate and potentially generate particles. So there should be the duration of these monitoring needs to include the equipment assembly. There should be alarms with a timely response. Is there a light in the room? Is there a notification from someone? Is there a person monitoring these total particle systems? Now, obviously, a viable monitoring, you can't have a timely response because you don't get the results until later. Although there is a desire to have more rapid microbial methodologies, but not everyone has those. But the idea is any alarm system should be telling the operator as soon as possible, and so that some correction could be made. Do you isolate a certain portion of the lot where this event occurred? Do you stop certain activities? Do you allow the room to come to some new state? All these need to be laid out in SOP, and there needs to be a clear alarm. There needs to be an indication that this monitoring is being taken seriously. Grade A is to be monitored continuously for total particles. Again, it's noted at least 1 cubic foot per minute is pretty typical, and viable particles. So that means you need to put, the plates have a certain length of time, both settling plates and active air plates have a certain amount of time. Those need to be appropriately replaced so that you're continuously monitoring during the equipment assembly and the entire operation. Now, a similar approach should be used for grade B. However, there's less risk in grade B to aseptic process. So we will decrease the sample frequency and decrease the location. But again, you should look at your risk assessment in terms of where these samples are taken and where they're done. If you have a particular location which maybe in grade B, but it's where it enters the grade A area. Do you have a sampling location there? Because that's a higher risk of bringing particles into the grade A area. It is notable that viable particulate monitoring should be performed also when operations are not occurring, right? So we don't need to just have our variable particle monitoring within the operation, but also post disinfection, prior to the start, and so we talked about equipment assembly, completion of the batch, and after any shutdown of the clean room. Okay, now that we covered clean room and clean air systems, let's talk briefly about the sterile filtration section. So this section has been substantially expanded. It contains a lot more detail than it did previously. So if you're doing sterile filtration, it's important to read this thoroughly. There is now a requirement for bioburden samples to be taken immediately prior to sterile filtration. So the expectation is that even though you've probably qualified your system, your sterile filters to filter out a lot of bioburden, you still need to take that sample and monitor that sample. This is probably one of the more interesting or controversial aspects. Currently, the FDA doesn't require this, but the EU now has added this. The pre-use post-sterilization integrity test. That means that after it's either autoclaved or gamma sterilization, but prior to the use in the process, you have to do an integrity test. Now, this is understood. It is technically challenging, particularly if either the wetting fluid is not the product solution, right? So if it's not the product solution, then of course, after your integrity test, there's some flushing or removal of that wetting fluid that needs to be done before you can collect the product, sterile filter product. Or if the batch volume is particularly small. The reason this has been continued to be argued is that many feel that the implementation of trying to do integrity testing, essentially since it's post-sterilization, you're frequently now in a clean room, you're in an aseptic environment, trying to perform the integrity test, that implementation of that test and how you do that test may be riskier to the sterile filtration setup than actually the probability of having what we'd call an undetectable pre-use integrity test failure, right? So essentially we're talking about that integrity is lost prior to the sterile filtration process, but then somehow on the post-use the integrity failure is not detected. Now, there's only very specific situations where that may happen, where either the processing conditions or the product itself may plug the filter or distort the filter in some way, so that even though the filter shows as integral at the post-use, it was not integral during use. So that's where some of the concern has come from that has driven this PUPSIT methodology. You can provide an exception and say, this is why we cannot do PUPSIT. You can put a risk assessment together. Where does that risk assessment go? It's got to be connected to your contamination control strategy. You have to talk about what controls are in place of the filter sterilization process, how it's being sterilized, how the filters are being handled, packaged, whatever. If you're simply buying a gamma radiated filter and it's being just shipped to you, you really don't have a lot of control over the shipping conditions in which you're receiving it. And so, are you concerned that that filter could have been damaged, even though you're relying on an integrity test that was done prior to that, and the overall supply chain? What are the process conditions that it could impact filter integrity during use? I mean, do you have a process that could gel or plug that filter during its use and basically hide the fact that, in fact, it was not integral when it was being used? So essentially what you're doing is you're obliterating the chance of a post-integrity test to be able to detect it. And the overall risk of failure that you've determined to detect a non-integral filter post-use, right? So what flushing or methodologies you're using post-use to potentially recover that filter, and assure that a post-use test has higher probability of detecting that failure. So PUPSIT is required, but there are exceptions you can put together. They're trying to make it difficult for you to put the exception together, but you can put an exception together. The whole sections on barrier systems has been very much more reorganized and clarified. So it's broken up into two different areas: isolators, both open and closed, and restricted access barrier systems, or RABs. So the Annex 1 makes it clear that an open isolator, that's an isolator that shares air with the surrounding room. Obviously, it's grade A, unidirectional within that isolator, its critical zone, but its background has to be a minimum grade C, and you have to show through airflow patterns that there is a communication between air in an open isolator. You have to show that those airflow patterns do not ingress, obviously, into the critical zone. Now, if you have a fully closed isolator, you have to have grade A within that isolator, but you don't necessarily have to show unidirectional flow if your operations are relatively simple. Right? So if you're doing something that's very simple, doesn't have a lot of airflow disruption, you don't necessarily have to show unidirectional flow in a closed isolator. And the background is a minimum grade D. There are expectations of glove integrity tests at defined intervals. So you have to define that in your SOP of how often you do your glove integrity test. But it also requires that you visually inspect the gloves at each use, particularly for an operation that puts risk of potentially damaging those gloves. For closed isolator, the isolator integrity has to be tested at defined intervals. And there's an expectation that the sporicidal biodecontamination is an automated process, and that it is validated with defined cycle parameters. Restricted access barriers, of course, are more risky. So obviously the critical zone must be grade A unidirectional, but its background, because restricted access barrier systems actually are in fact opened, particularly to do aseptic assembly and such. So you still have the requirement of a minimum grade B background. You must show the positive airflow from the critical zone to the background, right? So it's an open system, so you have to show that the airflow is pushing out of the grade A area into the grade B area. You must show those smoke studies, those airflow patterns, to make sure that there's no ingress coming back in from the grade B area. If you have gloves on the RAB system, they must be sterilized or decontaminated for each campaign and sterilized prior to installation. And there should be a routine sporicidal disinfection, but it's not requiring it to be automated for a RAB system. These are new sections that have been added that are becoming more and more common use. The single-use systems, or these bag type systems, and a separate section on what they call closed systems. Closed systems are more commonly used in areas like radiopharmaceuticals and things like that, where there's particular shielding and things like that. For a single-use system, it is recognized that there are specific risks of using a single-use system versus a rigid system. So you really, in your contamination control strategy, have to recognize what those risks are and what you're doing about those risks, right? I mean, yes, you have a single-use system, and maybe that's very useful to you, but are you really defining what those risks are versus your typical rigid tanking and piping systems, right? It's very important to understand the sterility assurance and qualification of suppliers and sterilizers. It is not sufficient to just simply say, I bought it off the shelf and they sent me a spec that says it's sterilized. You need to understand, are they doing, if they're gamma sterilizing it, are they doing the quarterly dose audits? How does their qualification of sterilization apply to maybe your specific configuration? I mean, because you can configure your bag differently, and maybe it's significantly differently configured than what actually is being qualified as sterile. So you need to understand that. You need to understand those sterilization documentation to be provided by those suppliers. You need to understand compatibility. You need to understand extractable leachables. You may have to do an extractable leachable study to show what you can get out of these different single-use systems. They are designed generally to be compatible, but you need to understand with your specific drug, is there any aspect of my drug in terms of pH or whatever that can potentially get an extractable or leachable out that could be toxic, right? Obviously, your exposure time is a lot less, so it's somewhat simpler extractable leachables compared to a container closure integrity type, but still certain requirements. And this also includes elemental impurities, so don't forget that. This is a challenging one, maintaining that integrity throughout the processing. I mean, how do you receive these? How do you store them? Do you check the integrity prior to its use? Do you check the integrity after its use? Do you know these bags are maintaining integrity? Are they developing holes? Are they getting pinholes? Is there in fact potential issues there? So, and then of course all the assembly and connections. Even though you may have these intrinsically connections, you need to kind of show through your aseptic process simulation that you can do all these things and not somehow introduce any contamination within your systems. More briefly about closed systems. The real advantage of closed systems is it minimizes contaminations of particles and microbials, right? You have a completely isolated, sealed system. Design and selection of these systems to assure sterility of all the contact surfaces that are within these systems. You need to have a system pressure integrity testing performed regularly. And generally, you're going to be using these what are called the intrinsically sterile connection devices. If there's some risk to integrity, or you periodically have to open these closed systems, then unfortunately, yeah, it's a closed systems, but your background should be a grade A. If you don't have any risk to this integrity, you can justify it lower grade. Again, this is something you need to define in your contamination control strategy. So there's a lot of things we've talked about in terms of what we need to implement within by August. There are a lot more things within this document which we haven't even discussed here. So the deadline for implementation is the 25th of August of this year. And for the specific section on the lyophilizer loading, it's next year. So what do we do about it? So the first thing you really want to do is you want to go through and find those particular revisions that are relevant to your systems, your whether they be sterilization, aseptic processing simulation, your clean room design, your facility design, your gowning, whatever. Perform that system level gap assessment. Determine where do I have risk? Where do I have gaps? Right? How, if I have that gap, how big is that risk? You definitely, in addition to that risk assessment, you definitely want to try to get that draft done of that contamination control strategy. Bring all those pieces together. Have something that says this is our contamination control strategy. This is all the different pieces and control aspects that we have in place. If in fact you have a new facility product process or any sort of system launching after, I don't mean by launching after August last year that it would be compliant, fully compliant by August of last year. It needs to be compliant by August of this year. But if in fact you've launched that new thing after August of last year, I think that it'd be reasonable that they would expect that within a year that you'd be compliant in that new system to the new Annex 1, right? The reason they believe that, although there was argued by industry that they needed a 2-year implementation was requested, because of the delays of actually getting the final version out, and because there were drafts out for a rather substantial length of time, the EU decided that they'd given enough information to industry that they knew what was coming, and they really have an expectation that within a year that we should be able to do that. Now, of course, existing facilities, that's really what we're dealing with. I think it's a challenge for industry to take some of these aspects and get them particularly related to things that are now included in terms of facility design and things like that, to be able to retrofit and things like that within a year. That's probably a little bit beyond reasonable expectation, but put together a plan. Take that risk assessment. These are the areas they feel are higher risk to contamination control. This is our plan. Okay, we've identified these areas based on the new Annex 1, and we have a plan. This is when we're going to put things together. I think that's the best you can do when you just don't have the time to implement everything. There's a lot of different changes, a lot of different systems that are impacted here. A lot of these particular changes, the EU views that some of these changes were best practices anyway, and that you should have probably been doing them. But we all live in the real world, and we can always find gaps with our just various systems that are in place. So best you can do, get that contamination control strategy done, have a risk assessment, put a plan together. So at least you can show that to if you have any sort of audit happening. We don't know what areas the EU will focus on, what areas will be the most aggressive audited areas. We won't know that until those audits start happening after August, and then we'll see, and the industry can appropriately respond. This is my contact information. And let's see what we do for questions here. Okay, if I perform a sterility test in an isolator, should the surrounding area classify it or not? And which class should it be? The Annex 1 is not specifically talking about sterility testing in terms of isolators. I would expect that they view an isolator as an isolator. So depending on whether it's open or closed, the surrounding environment, if it's an open isolator, minimum is grade C for the surrounding area. If it's a closed isolator, minimum is grade D. Remember that grade D in operation, you determine by risk assessment what limits it has. So you have that little bit of flexibility there. How to evaluate the risk in QC labs when performing GMP on the manufacturer. I don't know the specific requests, what this question is about. The risk of QC labs performing GMP. I think this is a broader question, maybe than just Annex 1. If you're talking specifically about Annex 1, Annex 1 is not specifically talking about the labs. If you're talking about micro labs, obviously micro labs that are performing certain things like sterility testing and microbial testing, if there's a clean room applied, then yes, Annex 1 does apply. It is about maintaining contamination control and such. I'll have to think a little bit more of what specific aspects of this question are required. How to check the qualification isolators and biological indicators for hydrogen peroxide, and should this be in a routine testing? Certainly, the biological indicator methodology for the qualification of the decontamination process of the isolator is a requirement. The frequency of that testing, I'm not familiar enough with this to tell you whether it's required. I would expect, like all decontamination or sterility type of tests, that an annual requalification would be appropriate, mostly to assure over time. I don't know if that is a specific requirement. It's probably good practice. I mean, it is a decontamination, it's a sterilization process. Things could happen over time. Gamma, we do quarterly, autoclaves, we do annually. All these different requirements in the sterilization process have requalification, so I would expect that requalification would also apply here. Pre-use filter qualification, single-use sterile filters puts the sterility filter at risk and does not add safety. As opposed to integrity, include anything that may have happened during fluid sterilization. Frequently filter things a QC managed test outside the clean room, as does not make sense. Yeah, this is I would agree. PupSit has been very controversial. I'm not really a fan of PupSit. I've seen very few processes that would benefit from this. The only particular process that I saw that would particularly benefit from this, assuming you would have a integrity problem pre-use that you would not be able to detect post-use. I did have a product that basically what it would do, it would gel in the filter, and it was very difficult to flush out that product from that filter. So you could make an argument. I'm not saying that there was an integrity problem, but you could make an argument that you would not be able to detect an integrity failure post-use for that particular type of product. So that might be a product that might be more difficult to do. Now, of course, a lot of us are using simple water-based solutions. They don't have high concentrations necessarily, don't have viscosity issues, things like that. They're room temperature. My expectation is draw up as defined in the Annex 1, draw up the risk assessment saying we do not feel this has any value to what we do, and basically put that in your contamination control strategy. I think that will continue to be a very For a classification table one does not have specific grade B at rest 5 micron, how to interpret the results for initial classification. I think that the way you can interpret those results is look at the monitoring table of what the monitoring. Now, remember, you can set it up in the initial classification so that the grade B 5 micron is such that you wouldn't necessarily fail, but you definitely don't want to go into monitoring with that 29 particles per cubic meter and basically constantly be failing either. So, what's the point of it's kind of weird that the way they put it together is that, like, oh, yeah, you don't need to classify it, but you definitely need to monitor it. As we understand, of course, during the classification, we want to understand we're going to pass our monitoring on a regular basis. So it really kind of depends on how you set up. How are you going to monitor that 5 micron? Like I told you, the difficulty of the 5 micron with 29 particles cubic meters, you're measuring at a cubic foot per minute, which means it's the limit in 1 minute is 29 divided by 35.2, which of course is a fractional number, which doesn't make sense. So you can't look at it every minute. You have to look at it over a couple of minutes, and yeah, you're literally dealing with looking at for single particles in the measurement sample. That's really hard to do with an electronic instrument because you can just have electronic noise and you'll see a particle that's not there. So it's real. I agree. This is really going to be still continue to be challenging. The next one, what about disassembly and non-viable particle monitoring, as it makes complete sense to do that? It does not talk in Annex 1 about disassembly. We understand that obviously disassembly may generate more particles. I've seen that argued with data where they basically said, oh, yeah, we had a particle spike, but looking at the timing of it, it was occurring while we were doing a disassembly. Frequently for things like RABs, you're going to have those RABs doors open to be able to do those disassemblies. I think what this is one of the things you make very clear in your CSS, CCS, is that, yeah, you are not going to be doing this during disassembly. Maybe you will do it after the disassembly is over, right? Mostly just to make sure that, like, okay, yeah, what does my clean room look like? Did I contaminate my clean room during disassembly or such? But like I said, that's what that contamination control strategy is for. Old Annex 1 employs the term limits for microbial monitoring, while Annex 1 employs a microbial contamination level. So word level imply rough limit. Around the number of microns tagged into the fifth CFU for grade C can be considered. Yeah, well, yeah, there's some interpretation because we all know that CFUs are not a precision number. Yeah, I'm not a microbiologist. I've had this discussion before. I'm not really the expert in this area of whether 105 CFU really is over 100 CFU. Again, this is also why you want to have alert limits that are actually probably below. And it like any event like this in an investigation, if it's isolated, if based on its location, maybe it doesn't have a high risk. Just going over level doesn't mean, oh, fail, 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 reject, reject, reject. It means investigate. And investigation may determine, say, listen, this is not based on trending, based on whatever, based on the accuracy of micro methods. This is really not, 105 is not really much different than 100. So, okay, what do we do about it? Well, okay, maybe we'll resanitize the area, we'll resample the area, it doesn't come back. Now, if it happens a lot and repeats and repeats and repeats, that's a different story. And that's the reason why we do trending.